Hello.
Good morning.
Good morning. Good morning, Anil.
Good morning.
Yeah. Good morning. Very good morning. Has the speaker joined?
I don't know. Sunil only knows about that.
Sunil told that he would be joining soon.
Okay. Okay.
Sunil, can you hear me?
Yeah, I can hear you.
Sunil. Yeah, Professor. Professor Chary, can you unmute that? Yeah, very good. Very good. Yeah, very good. Very good.
Wonderful. Thank
Wonderful.
you.
So probably you can upload the presentation right now before we start.
I will do that.
Okay. Thank you, sir. Sunile. Sunil.
Are you able to see that now?
Yeah, it's possible.
Is it clear?
Yeah, clear.
Wonderful. So you just prompt me when I have to start, I will do that. So.
Yeah, we can start, but before that, we will have some introduction. Did you upload that, sir?
One second, just one second. Are you able to see?
No, not yet.
I have uploaded.
Yeah, but did you share that?
I can see.
Yeah, I did share.
But the screen we see is really the. Can see that?
Yeah,
I can see that.
Yeah.
Okay. Wait, let me check. Okay. Yeah, very good. It's working. Yeah.
Yeah. All of you, how many of you are viewing this?
Oh, right now 131. Actually, there are more than 500 students registered.
Okay.
Yeah, but right now 134, 35.
Okay.
Yeah, 135 or something.
Wonderful. So this is going to be 2 hours?
Excuse me, sir?
Is it 1 hour or 1.5 hour? 2
Yeah,
hours?
1 hour. Then we have some discussion. We will take some questions from the participants, and then we can.
Fine. Perfect. 1 hour.
Yeah. Then okay. Sunil, can you start right now?
Yeah, please.
Okay.
Yeah.
Hello. Welcome to all to the webinar series of Nmr workshop, Nmr theory, instrumentation, and application. I am Dr. Arun from Kerala State Council for Science, Technology and Environment, which is a Science and Technology Department. It's an institute under Science and Technology Department of Kerala government. And we have several programs for the promotion of science education, and popularization of science. Several schemes for benefits from school children to emeritus science. And besides that, we have 8 R&D institutions under KSCSTE and SRIBS, Srinivasa Ramani Institute for Basic Sciences, one of them. We conduct several capacity building programs for PG students. And this is third in the webinar series. And we had first one immunology and immunotherapy program webinar series. And then we had catalysis two weeks before. And now we have Nmr webinar. And Dr. Sunil and Dr. Anil from University are the coordinators, academic coordinators of this program. And NMR, as you all know, that is one of the powerful analytical tool in chemistry, not only in chemistry, but in physics as well as biology. If you look at the Nobel history, you can see almost 8 scientists were awarded Nobel Prize in 6 occasions, starting from Otto Stern, that was 1943, for the magnetic, he discovered the magnetic moments of protons. And then Isidore Rabi, who recorded magnetic properties of nuclei. And 2003, 2 Nobel laureates were there for MRI, magnetic resonance imaging. So this is a wonderful technique for many scientists, actually. It's very sophisticated. And because of the development of high computer capacity of computers and all, and Fourier transformation is possible now, and NMR is now it's very simple for, you don't need much technical expertise to do that. So I'm sure that in these 4 days of workshop, we will have some idea about how this is done, what are the instrumentation, and we have very good experts for delivering, handling the classes. With these few words, I invite Dr. Sunil Kumar for introducing the speaker. Thank you. Dr. Sunil?
Yeah. Good morning, all. Good morning, Chary sir. This is Sunil.
Yeah.
So, we are starting with our first session of webinar on NMR spectroscopy. So, our first speaker is Professor Chary. I am very happy to introduce and welcome him to this webinar. I know him personally for the last 19 years. When I was a doctoral student in MG University, I was a frequent visitor to his laboratory for doing NMR analysis. And Professor Chary trained me well in NMR, and he helped me throughout my career. Professor Chary obtained his PhD from Osmania University, Hyderabad, and joined TAFR Mumbai in 1983. He was a visiting scientist in the laboratory of Nobel laureate Professor Kurt Goodrich at the Institute of Molecular Biology and Biophysics, ETH Zurich, during the period 1988 to 1990. Thereafter, he served at TAFR in various capacities, including as the chairman of the Department of Chemical Sciences and was most recently a senior professor. Professor Chary is a renowned expert in NMR spectroscopy, and over the last 35 years, he made tremendous contributions to several diverse areas in structural biology, molecular biophysics, and biological chemistry. In recognition of his scientific contributions, Professor Chary was awarded the G.N. Ramachandran Gold Medal for excellence in biological and sciences and technology in 2017 by the Council of Scientific and Industrial Research. In 2017, he was entrusted with the responsibility of nurturing and navigating the new IISER campus at Berhampur as its founding director. Professor Chary published several hundreds of research articles in journals and some books related to NMR spectroscopy. He guided many doctoral students and postdoctoral students. He was the secretary of ICMR and presently he is the secretary of NMRS India. I heartily welcome Professor Chary for his talk on the topic Esoteric NMR spectroscopy.
Thank you so much, Sunil, and Arunan and everybody at your end for inviting me to give this talk. And I chose a very specific title, Esoteric NMR Spectroscopy. I'm sure most of you are aware of what NMR stands for. Am I audible?
Yeah, audible.
Yeah.
Yes.
Yeah.
So, before the start of this talk, I would like to show you just a few slides of the IISER Berhampur. So we I have just moved from west coast to the east coast, and we are right on the Bay of Bengal shores, and this is our boundary. Just 60 meters from the seashore, we have a very thick forest-like site of 200 acres of land, which is full of cashew nut trees and coconut trees. And we are going to transform this into this kind of a beautiful campus. And what you see on one side of the road in between is the academic block. On the other side, you have hostels and the residential complex. So from the high tide, I mean, our boundary is just only 60 meters. It's a virgin beach. Everybody who has visited the site was wondering whether the students and the staff will be sitting in the academic block or will be spending all the time swimming in this sea and enjoying the beautiful seashore. So this is eventually what is going to be the academic block. Under one roof. We'll be housing chemists, biologists, physicists, mathematicians, and computer scientists, and even other branches that will evolve with time. So having said that, I come back to Eric. NMR. NMR stands for nuclear magnetic resonance, a resonance phenomena which is due to the magnetic property of the nuclei. When I say nuclei, it's not that all the nuclei are NMR active. NMR was usually, comically, were to be identified as no more research. But every time somebody says that there's no more research, people working in this field have been proving that there's a lot to come and lot to come. NMR is an intricate and elaborate choreography of certain nuclei which have a spin off or more than spin off. For example, I have listed a proton, hydrogen nucleus, or deuterium, carbon 13 or N15 or P31. All that I have listed here are the most often which are used in the study of biomolecules, which are going to be the subject matter of the today in the latter part. These nuclei respond with mellifluous melodies, which are not palatable to human ears, but carefully picked up by electronic ears to decipher their songs composed in the language of radio frequencies. If you just put a water in NMR spectrometer, what you see is a single line which I show here. If you take ethyl alcohol, what you see here is the three lines here. One coming from the OH2 proton, another one coming from CH2 proton, another one coming from CH3 proton. If I horizontally expand, you would see the multiplicities in this, which has a lot of information embedded in it, and that's called coupling constants, rather which indirectly provide you about the stereochemistry of the molecule. And now, if I put a protein dissolved in water in the NMR spectrometer, what you see is this kind of spectrum. This could be a representative of 700 to 800 spins. What it means is that you have a spectrum which is frequency versus intensity. When I say that this spectrum is a signature of 700 odd nuclei, you mean to say that, what I mean to say that is that it indeed has 700 signatures. There's a wealth of information embedded in this spectrum. And the art of getting that embedded information is what made this NMR a very, very powerful tool today. I just wanted to highlight, in introducing the subject, somebody was talking about there were 8 Nobel Prizes. It is a wonderful journey since 1945 to today. First, it was bulk materials by Bloch and Purcell, who were basically physicists. And in 1950s and 60s, it turned out to be the tool in chemistry. The first one to appreciate was the Gutowski and others. I should also highlight that there was a contribution of one gentleman called Dharmati too, who was the first person rather who has found this so-called chemical shift. I don't want to go through all this list, but want to highlight that in the wonderful part happened in 1966 when Professor Ernst presented what is known as FT-NMR, a single pulse that can excite and all present in this sample. And thereafter, from chemistry, it even went to medicine and even what we popularly call as functional MRI. For more details, I think I would advise you to read a book which we have published in 2008. It's called NMR in Biological Systems, From Molecules to Humans. So the realm of NMR, you can contribute immensely in developing new methods. It could be used to address various problems in structural biology. You can use this technique not necessarily in water or in aqueous solution or in any organic solvent, but it can also be studied in solid state. And the contribution to this field can be done even by computer scientists in developing some software and hardware. One can even see how a metabolic activity is on in a given organism by making use of this metabonomics. Or you can characterize various materials and organic systems. And as I told you, you can use it to image internal organs of a body. That is what is popularly called as MRI. Or it could be even studied to see in real time what is happening within a body, and that is what is known as functional imaging. For those who have not seen Nmr spectrum, I would like to show you the first Nmr signal in Paraffin. This was published by Professor Philips Bloch. You would wonder how this kind of signature still could win a Nobel Prize in 1954. So these are the two people who have contributed to that. One on the west coast of America and the other one on the east coast, and from Stanford and from Harvard, who had bestowed this Nobel Prize in 1954. And this is what I highlighted, the contributions of an Indian called Dharmati, who is the father of NMR group in DFR. He was the first person to show that ethyl alcohol not necessarily gives just one signature like that, but instead it gives 3 signatures, and shown that is due to It's the chemical environment of each and every spin that is present in this molecule. And the moment a chemist started realizing the importance of this NMR, the biologists were no longer behind, and they took a protein and tried to record as way back as 1957 and realized that it indeed gives a wonderful spectrum with just 4 shoulders. And they could account the signal 1, 2, and 3, and 4 indeed corresponds to the number of protons that are supposed to be resonating at that place. So that was the interesting thing actually. And it inspires a young mind how to look into a spectrum. To start with, it may not show any signatures or any usefulness, but the thing is, over the time, people realize that this kind of spectrum could be translated into this kind of spectrum. In 1995, Signal protein, which had 700 protons, recorded in 600 MHz spectrometer, showed this kind of thing. And this was possible only just because of the contributions of Professor Ernst, who has conceptualized the technique called FT-NMR. Now, I change the gear and try to illustrate how one can derive a structural information from this kind of a complex spectra. Why study the structure? It is in the structure and in the dynamics. Understanding the structure of a protein is important for understanding both its biology and chemistry. And once you have a novel structure in hand, you can make it a cover story, either in Nature or in RNA or Science, as I have exemplified in this cartoon. Now, quite often I am asked what are the kind of sample requirements. And when it was shown the signatures of the water in, people were thinking that it's only you need millimolar samples. But today, you can even record the spectra with micromoles or nanomoles. Size limitations have also changed. It started with 18 Dalton water molecules today, but studying even 1,000 kilo protein or protein complex. And the secret of this success has been primarily because of NMR applications to biological macromolecules. What are these biological macromolecules? Most of you might have already been introduced to what is known as the DNA, RNA, and proteins in either in 12th or in graduation. These are a large molecule.
Excuse me, Professor Chary.
Yes.
I think the sound is not clear. There is some echo. Is it the audio cable is okay there?
Yeah. I'm within.
Voice is not that clear. Sometimes it goes up and then come down. Sometimes echo. I think the cable may be loose or something. Can you check that, please?
No. I'm completely.
Yeah, now it's perfectly fine. Now it's perfectly fine.
I have kept the closer now.
Okay, yeah.
Yeah. So you have DNA and RNA and proteins. These are the macromolecules. Are my slides visible clearly?
Yes.
Okay.
Perfect.
Right. Now, I just introduced before talking about the structure and dynamics of these wonderful molecules. I may not be able to have time to discuss all the molecules, but at least I will be talking about the proteins. Now, before doing that, you should all know this central dogma that DNA, the genes that are present in our DNA, transcription takes place into RNA, which gets translated into protein. Okay, So these are the three important, very, very important macromolecules. Okay, a DNA is a string of nucleotides made up of adenine, cytosine, guanine, and thymine. On the other hand, RNA, which is DNA's cousin, has adenine, cytosine, guanine, and uracil instead of thymine. And proteins are made up of 20 different pearls called amino acid residues, which are identified with single letter codes, A for alanine, C for cysteine, and so forth. Y stands for tyrosine, W stands for tryptophan. Okay, so there are 20 different kinds of natural amino acid residues which make these string of amino acids. And any given protein sequence is represented by a primary structure. A primary structure is nothing but simply a sequence of single letter codes, each one representing an amino acid residue. These primary sequences end up folding into beautiful alpha helical beta sheet structures called secondary structure elements. There are also beta bends, gamma bends, and other secondary structure elements too. And these secondary structural elements end up into giving this kind of a beautiful tertiary structure. A calcium binding protein which has two domains, a single long polypeptide chain folds into 8 helices, 4 beta strands, and several loops, and gives very beautiful tertiary structure. It is not always true that these structures are very globular. Sometimes a given protein may have very ordered structure at the C-terminal end, but its internal stretch may be very, very flexible, as it is in this case of a prion protein. On the other hand, you may have a different kind of a protein rather where somewhere in the middle it is very flexible, but the rest of it is all folded. The N-terminal and C-terminal segments. So during the last 3 decades, 3 significant developments took place. One is the sequencing of various genomes and development of NMR spectroscopy, and more recently cryo-electron microscopy. I'll just try to highlight what exactly it is this. You may be knowing from the day the human genome has been sequenced, there are several 300,000 genomes have been sequenced. It's growing exponentially. On the other hand, number of protein structures that are solved also have been exponentially growing, as I have exemplified in this slide. So this is the way rather you are in an exponentially increasing world actually. The amount of the data that is coming out of these human genome projects are enormous. That means that if you have a genome sequence in your hand, you have wealth of information. You know all the genes that are present in that given genome. The question that you would ask is that, what are all those genes and what are their functions? To understand that, it would take a long time. Now, as I told you, the exponentially increasing number of depositions in the PDB more is the one rather which I'm highlighting here. Electron microscopy is also catching up. So what it means is that although the number of crystalline NMR structures deposited each year in the PDB has been growing exponentially, the number of structures unrelated to other proteins has not. So when I say that there are so many structures, okay, it doesn't mean that each one is unique. No, certainly not. This is due to the part because of the practice of the structural biologists, looking ever deeper at different biological macromolecules, studying complexes, mutants, and relatives of the proteins of already known structure. So, it is also, one should appreciate that the universe of the protein structure is definitely finite. Not necessarily that every day you find a new fold. The chance occurrence of structural similarity have become increasingly common. This trend would appear to be strongly positive for the task of deriving function from these structures. So the message is that the rate of generation of the new structures ought to accelerate to even start keeping up with the sequence information being generated now. So this is where NMR is gaining importance in characterizing the structure and dynamics of the proteins and nucleic acids, and most importantly, in solution as well as in solid state. It is complementing results obtained by X-crystallography. 1/6 of the new structures in the recent times have come from NMR studies. The basic problems in NMR are sensitivity, resolution, and water resonance, sequence-specific resonance assignments, and instrument time. I have seen other abstracts. I think other lecturers you have chosen are going to highlight and may take you much more deeper. So this being an inaugural lecture, I'm just very superficial. But if you're not able to understand anything, rather, please come back and stop me, and I'm there to take your questions, okay? So when I say that the problems are sensitivity and resolution, this is what triggered everybody to say that, oh, NMR stands for no more research. But people have proved, and over 75 years, the total gain of the signal-noise ratio has increased enormously, 300,000 times. It is primarily because of the higher fields and high temperature superconducting probes and low temperature coils and preamplifiers. Better RF technology too. Again, we are in an exponential world. The sensitivity and by normal probes as well as the cryogenically cooled probes are also increasing year after year. And if you have a 1000 megahertz NMR spectrometer, you can have signal noise ratio anything to the tune of 10,000 with a cryo probe. Now I will switch to how we derive the structural information from a given spectrum. To understand this, I start with a single amino acid, and that's what I call it. I have chosen threonine. It's one of the 20 natural amino acids. And what you see here in red are non-exchangeable protons. So if you dissolve this threonine in aqueous water, so you may not see that OH and NH because they rapidly exchange. But however, you would see those three signatures. So I will just introduce you to the one-dimensional spectrum, which was the FT-NMR technique that was proposed by Professor Ernst. So what he did was that to excite and record the signatures of these three signals or three spins, alpha spin and beta spin and gamma spin, what all you need is that a single pulse of 90-degree pulse. Okay? And then after application of that, you see the response in the form of a freely decaying signal. And this has all that frequency information, and which is not easily observable unless and otherwise you transform this freely decaying signal into this beautiful spectrum where you see 1, 2, and 3 lines. And this one has a large multiplicity, and this one turns out to be a doublet, and this one turns out to be another doublet. The moment you see a doublet, that means that these protons are coupled to another proton, and even this proton should be coupled to another proton, whereas this proton must be coupled to more than one proton because it is further split. So by looking at vaguely about the multiplicity itself, you should be in a position to understand which spin is which. I have already identified that this is H beta because this is coupled to H alpha and CH3. So people depended on the spectral signatures to identify these. Now I want to switch the gear to this one-dimensional world to higher dimensional world. The seminal contribution for this transformation from one-dimensional world to the multidimensional world came from Jean Zenner, a Belgian physicist who has proposed that the magnetization belonged to a spin I could be transformed to spin S if I and S are coupled spins. When I say coupled spins, in an ethyl alcohol, CH2 protons are coupled to CH2 protons. So that means by this method, I can transfer the magnetization from CH2 to CH3 or CH3 to CH2. So what is needed is that the condition that I and S spins are coupled, and you need a non-selective 90-degree pulse which excites I spin and S spin, and I going to Z direction from X and S going from Z direction to X direction. So this kind of a transformation result in exchange of magnetization. So he was a theoretician rather. He was not able to use this technique, this idea, and as he was not an experimentalist, he couldn't show it experimentally. So for 3 years, from 1971 to 74, this idea was dormant. It was only when Professor Ernst opened up this Pandora's box and coined a new experiment called correlation spectroscopy, which is not just a single pulse experiment, but two pulse experiment, and see the response here, where you have two time domains. That means that FID is now a function of two time intervals. And what he did was that he created what is known as a preparation period, evolution period, and mixing period and detection period. And he created two time domains here, one directly detected time domain and one indirectly detected time domain. And by changing indirectly the time between these two pulses in steps of delta T1, okay? So he made this as a variable and this as a variable. So now the recorded signatures are function of T1 and T2, and two-dimensional pore transformation of that gives you a 2D spectra, where the diagonal is the one-dimensional spectrum and off-diagonal peaks have a wealth of information that was highlighted by Jean Zenner. To understand this, again, I go back to this threonine molecule, where I want to see the exchange of magnetization from alpha spin to the beta spin, because these are coupled, and from beta spin to the gamma spin. Can I? The question is indeed yes. The Cosy experiment proposed by Professor Ernst enabled the observation of these off-diagonal peaks where this magnetization has been shared or transformed to this spin, and this spin magnetization is transformed to this, primarily because the gamma is coupled to beta and beta is coupled to alpha. So by just looking at the spectrum, I'll be able to authoritatively say which spin is coupled to which. Are you able to understand this? Hello? Are you able to understand?
Yes, sir. Yes, sir.
Okay, wonderful. Okay, thank you. Okay, now I will make it a little more complex. A complex like this. If it were to be threonine, isoleucine, phenylalanine, arginine, the spectra would be something like this. But I can still identify, hey, this gamma of threonine is coupled to this beta, and this beta is coupled to this alpha. So I can see the signatures of the threonine in this complex spectrum, okay? And by spending another 15 minutes, I could have explained to you how to identify other amino acid residues presented in this beautiful tetrapeptide called TIFR, a unique one where each amino acid is appearing only once. That means that each amino acid is unique and as unique as TFR, and each signature is unique, and so it is easy for you to understand and appreciate what it is, and you're able to see that. But it is not so colorful, mind you. It is all black and white. But given little training, I'm sure every one of you will be able to identify that this one is the signature of threonine coming from gamma to beta transfer and from beta to alpha. Mind you, this is something like solving a quiz. Now, if this is the kind of spectrum coming from a 4 amino acid residue long peptide, what it would be? A protein. A protein with 94 amino acids will show up this kind of a complex signatures, and each dot of diagonal dots represents a live coupling between two spins, and the game is to solve the puzzle and identify all the couplings coming from 94 different kinds of amino acid residues. So this has been, and the moment the people saw this, people were breaking their head and saying that, hey, come on, you cannot derive anything out of it. And again, people were started saying that there's no more research. Now, I will just, to exemplify the complexity here, I'll just again go back to another simple amino acid called AMMA. Alanine, methionine, methionine, alanine. Now, this one is, though simple, there is a redundancy. Redundancy means there are 2 alanines and 2 methionines. And you know that each amino acid will definitely, both the alanines will give same signatures. So how do you identify which signature corresponds to the alanine at the N-terminal domain, N-terminal of this peptide, and the one which is at the C-terminal? It is impossible to find out. But there is a possibility that if you can instead identify as the dinucleotide, dipeptides, alanine, methionine, 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 alanine, you can easily distinguish which alanine is which. And this is where the contribution of Professor Utrecht came in collaboration with Ernst by using what is known as a Nosy Technique. A Nosy spectrum is a 3-pulse experiment, rather, which has again 2 time domains and 1 mixing period. which eventually gives this kind of spectrum. Now here, unlike in Cosy, in this spectrum, each signature is something coming from the distance between two neighboring spins. If the distance is less than 5 angstroms or 5.5 angstroms, I mean, you should be able to see a signature between those two spins. So what it means that this indirectly gives the distance information between neighboring spins. That means if two spins are too further away, they would not have this signature, but if they are very close by, you will have the signature. So from this kind of thing, now, for example, just imagine this hall in which you are sitting. If you know the height and width and breadth of the room, and you will be able to design the hall in a very like manner by looking at this short distance signatures in the Nosy spectrum. You will be able to get the molecular structure, and that is what precisely was done way back in 1980s in Uthrich's lab in collaboration with Ernst. And this kind of a development of going from one-dimensional to two-dimensional has provided a powerful arsenal of versatile tools, and there are a number of techniques that have been proposed during that time. And again, when people saw, some of them were very skeptical, saying that if this is the kind of the spectrum which is coming from 100 amino acid protein, that's it. You cannot solve larger systems. What I'm trying to highlight here is that if 100 amino acids protein is giving this kind of a spectrum, if you take 200 amino acid big protein, naturally look something like a blot on a paper, ink blot on, okay? So resolution of that kind of a spectrum is impossible. So people said that, again, no more research. And this is where people then thought that instead of having the protein, the proton-proton correlation, whether we can have any other kind of a correlations. And that is where people, in tackling larger proteins, larger than 10 kilodaltons, where there is an overcrowding of peaks, and as the molecule becomes larger and larger, it tumbles very, very slowly. When it tumbles very, very slowly, the NMR signatures coming from that protein decay very, very fast. So the NMR-based approach, Nosy-based approach for resonance becomes impossible. So there should be some alternate strategy. And that is where people started using C13 and N15, but unfortunately they were only in 100 was C13 and only 1 in 300 was N15. So there was a need for isotope labeling. What is isotope labeling? If you were to use a natural protein extracted from any organism, you would not be able to record a good C13 N15 spectra at all, because only 1 in 100 of the carbons are C13 and only 1 in 300 are N15. So there was a need for isotope labeling. Isotope labeling is a method developed by biologists in which you have this kind of a plasmid which has your protein gene in it, okay? And that gene, which is embedded into a small plasmid, is transformed into bacteria, a bacteria which multiplies within few hours. So by the time the recombinant DNA multiplies or doubles, these small plasmid DNAs multiply several fold. That means there is an overexpression of these plasmids which are present in the bacterial cells. So what it means is that because it has the gene signature of your protein, the protein expressed by this plasmid also will be several fold. So that is how, in principle, by just in the sequence of your protein of interest, today you can biosynthetically overexpress using simple bacteria. For example, E. coli is a bacteria which is commonly used, very, very cheap actually. And when you are multiplying this bacteria, what you do, you cleverly feed them C13 labeled glucose or N15 labeled ammonium chloride. So what happens is that the bacteria, unaware of what it is eating, eats C13 glucose and N15 labeled ammonium chloride, which are the sole source of nitrogen, and eventually all the protein that comes out will be now C13 labeled and N15 labeled. So you can fractionally label it or uniformly label it, okay? And at the end, what happens is that entire protein sequence will have all the nitrogens in the N15 mode. The fact that it's in N15 now is a spin-off nucleus, and now it becomes active in transfer of the magnetization between its directly coupled amide proton and N15. So you can use that Zener idea and Cosy proposed by Ernst, and eventually you record what is known as heteronuclear Cosy spectrum, which is a little more complex than the normal Cosy because it has to have two channels, and you decouple when you are recording the spectra, and you get this kind of signatures. One axis being N15 and other axis being the proton. What you see here is that this N15 spectrum is 100 to 130 ppm, is 30 ppm broad. That means there is a lot of dispersion. Usually, those who have seen Nmr spectra of proton Nmr spectra, you might have realized that there are either just 10 ppm or 15 ppm in their width. But what you see here, the large dispersion of the peaks because of this N15 nuclei. Okay? So if a given amino acid has 200 amino acid residues, you will have 200 signatures. The transfer coming from within an amide group from proton to N15. So that is the hallmark of this heteronuclear correlation spectroscopy, which resulted in resolving the signatures of each individual amino acid residue. Now, if I can transfer from proton to N15 and record this kind of spectra, I can as well do from proton to carbon-13. And people started even thinking of transferring from proton to carbon, carbon to N15, or proton to N15 and N15 to carbon. And this resulted in several dimensions. This gave birth to what is known as a 3D and 4D Nmr spectra. And way back in 1991, when I was with Utrecht, this was the first protein 3D spectrum which was recorded, which had 2 proton dimensions and 1 N15 dimension. This is the 3D Nosy spectrum, where now the complex spectra which I have shown is now dispersed along the N15 dimension into several, 30 ppm width N15 signatures. So all that resolution problem which we were facing at the time could be resolved by recording this kind of spectra. And not only that, this also gave birth to what is known as triple resonance experiment, where you use C13 and N15 doubly labeled proteins, and you can transfer the magnetization from proton to N15 and N15 to carbon. So this has led to what are known as triple resonance spectra. To cut short the story, you can transfer the magnetization from N15 to proton to N15 and N15 to C alpha protons, okay, of the self and the sequential one too, because these couplings are very, very active. So by recording this kind of H and CA spectrum, where H stands for amide proton, N stands for amide nitrogen, and C alpha stands for self and sequential, in principle, you can walk along the sequence from this C alpha to N15, and from this to the neighboring, and from there to self and sequential, and go on and so on and so forth. So you can as well limit your correlation only to the sequential and forgetting about the self. So that way you can distinguish which is coming from sequential and which is coming from self. So this resulted in what is known as triple resonance experiment, which is little more complex because it has now 3 channels: a proton channel, N15 channel, and carbon channel. That is the interesting development, and which resulted in recording a proton N15 carbon 13 correlation spectra. Now you just see that 134 amino acid, one frame of this, such a beautiful, highly resolved spectrum from where you can now derive the chemical shifts of proton, N15 and C13, which themselves have wealth of experiment. So in principle, by using any of these spectra, one can walk from one end to the other end, from C terminal to N terminal or N terminal to C terminal. However, people started still feeling that it may not be as simple as I am narrating it. So they have still proposed new techniques called CBCA-NH, where you even correlate to the C beta protons, the C beta carbons, okay? Or even sometimes you use the carbonyl carbon too. And what it means is that in order to identify the chemical shifts of all these spins that are present in this complex protein, you need to record at least 4 to 5 or 6 spectra. Okay? And now, to analyze these 5 to 6 spectra is a big issue. Manual assignment is not is not easy, is a tedious and time-consuming task. So then came an opportunity to automate it. Automate by using some computer algorithms. So what all it needs is a good input of peaks and reliable classification of that. And that resulted in developing a automated assignment procedure in our laboratory called Tata Pro, and which help us to automate these assignments. Today, so even a non-expert can record the spectra and get all that information. What all I have highlighted is only a few of the complex spectra. There are several other procedures. Procedures involved where use of as many NMR active nuclei as possible. It's not necessarily that proton and C13 and N15. You can use even deuterium also. One can use uniform or selective labeling or fractional labeling even. You don't need to do 100%. Sometimes you may need only 10% of C13 labeling. And you may do completely deuterium labeling. Sometimes you call it as stereo-array isotope labeling. Sometimes you concentrate on residual dipole couplings. And sometimes you measure pseudo-contact shifts. And these nice labeling techniques have given birth to unknown coupling constants across the hydrogen bonds. Nobody ever thought that the spins across a hydrogen bond are also coupled. And today, these trans hydrogen bond scalar couplings have wealth of information, and that helps in structural analysis. And many a time today, ignoring the protons, people just concentrate only on C13 and 15, and that gave birth to what is known as protonless NMR. On the other hand, there is what is known as reduced dimensionality NMR, because as people started recording higher and higher order spectra, people were spending too much time on the spectrometer. As you all know, we are no longer in a 5-day world. 5-day to 1 day and 1 day to 2020 games have started. In a very like manner, instead of spending 5 days in recording a 4D spectra, why not record a 2020 spectra and get all that information that are embedded in 3D or 4D into 2D? So this gave birth to what is known as reduced dimensionality NMR also. So all this because of the long spectrometer time. I'll just briefly highlight that and pass on, okay? So the spectrometer time is a big issue which I highlighted. So a 3D in principle all the 3D information can be gotten in 2D, a 4D information can be gotten in 2D and 3D, a 5D information can be gotten in 4D, 3D, 2D, okay? So what you record is that simultaneously or co-increment or joint sample the chemical shift evolution periods as first proposed by Atreya and Sapolsky. Okay, so here what you're doing is instead of having T2 and T3 in a conventional 3D spectrum, which are the 3 independent time domains, now you co-increment T1 and T2 together as T1, T1. So basically this kind of spectrum will be 2D spectrum and that will be a 3D spectrum. And compared to this, and this time will be much, much less. So instead of sampling all the dots that are present in a conventional 3D, you sample only the diagonal and still have all that information. So the one 3D which would have taken 18 hours would now take only 26 minutes. And that is how, you could overcome the instrumentation time and get all that information that is embedded in 3D and 2D. So what I tried to do here, I highlighted several methods, and each topic which I have highlighted in the last 2-3 minutes would take almost an hour or two to understand each and everything. I would again refer you back to my book, which I have shared with you at the start of my talk. So now I will change my own interest actually in solving the structures and looking at their structure and dynamics and trying to correlate their function. Okay, I'll spend another few minutes, and in this, we were involved in developing new methods and studying the structure and dynamics, and we addressed the protein folding, and we did some experiments in cell NMR, and also we developed NMR software too. So I'm highlighting here a class of proteins called crystalline proteins, which make our beautiful eye lens. The transparent mammalian eye lens is made up of proteins called crystallines, which are all classified into 2 distinct superfamilies called alpha crystallines and beta gamma crystallines. Alpha crystallines are a member of a small heat shock protein family, whereas beta gamma crystallines are called structural proteins, they don't have specific, they are very, very high in density. Alpha crystals are 800 kilodaltons, beta and gamma crystals are 20 and 21 kilodaltons. And because of their sequence identity and sequence similarity, alpha and beta and gamma proteins, crystalline proteins are usually called as beta gamma superfamily, beta gamma crystalline superfamily. These crystallines are present in high amount, 450 milligrams per ml. And that's what makes our eye lens so beautiful and so crystalline. And mind you, it also has high concentration of calcium. How was this crystalline evolved from lower organisms to higher organisms? That was one of the questions rather which we have posed. Did the oldest organism on the planet Earth, for example, Archaea, has any signatures of the crystallines? That was the question rather which we tried to pose. And we also posed these questions like, why do lenses require such fairly high concentration of calcium? What is its utility? Do crystallines and what are the calcium binding sites, binding motif, and the conserved sequence of the calcium binding? Do crystallines oligomerize? And all these things actually. This is what made us to put all our efforts. The interesting thing is that if I have to address the first question, which I was saying that whether how this crystalline evolved from lower organisms. So when an archaea was sequenced, we tried to see in archaea whether this kind of sequence exists. This is the sequence signature of a gamma crystalline protein, where you have F at the first position, and at fifth position or sixth position, you will have a tyrosine or phenylalanine, and at I plus 7th, that is 8th position, you will have glycine, and somewhere between 28 and 36, you will have serine. So this is the kind of signature, rather. If you have a genome of archaea, and now go on searching whether you have this kind of signature. So in principle, yes, you can, okay? Now, before going to that, this kind of signatures can give rise to what are known as Greeky motives. A Greeky motive is like this. This Greeky motive has evolved over the time by gene duplication into a single domain double Greeky motive, and further duplication and fusion has resulted in 4 Greeky motive, 2 domain complex. And this is what exactly today's beta crystallines and gamma crystallines are, each one having 2 domains and each domain made up of a Greeky motive. A Greeky motive is nothing but a beautiful fold where the polypeptide chain folds into some kind of a beta sheet, actually, beta sheet structure made up of 4 beta strands. And now, coming back to my question, first of all, in a archaea, we tried to see the signatures of this sequence, and indeed we found, and then we studied its structure and ended up coming up with such beautiful structures, which indeed had these Greeky motive structures in the presence of the calcium and in the absence of the calcium and in the presence of the calcium. And this led to describe the first time the structurally closest member of beta gamma crystallines from archaea. This archaeal protein was shown to be ancient single domain beta gamma crystalline with its uncanny similarity to mammalian lens crystallines, a result of divergent evolution. The study also demonstrated for the first time that beta gamma crystallines are present in all the 3 kingdoms of life, thus making it the most prevalent, widely distributed calcium binding superfamily in nature. And then afterwards, we studied another protein from a marine bacterium called Hahella. And this surprisingly was ordered and disordered. Disordered in the absence of the calcium, but ordered in the presence of the calcium. And that gave a very nice opportunity to demonstrate how to study ordered as well as the disordered structures. And Hahellin was shown intrinsically disordered in calcium-free form, but undergoes drastic conformational change and acquires a typical beta-gamma crystalline fold after binding calcium, unlike other beta-gamma crystallines. Mind you, when there is no structure, you end up in such complex situation where the dispersion disappears and everything collapses into a short window of chemical shifts. And such a study of order to disorder, disordered order made a nice cover story. And then most recently, just a year back rather, we tried to look at the structure of a variant of human gamma crystalline that causes severe infantile cataracts. It's a protein by single point mutation at 57th position, a G being replaced by W, a glycine amino acid residue replaced by a tryptophan causes such severe infantile cataract. Again, we cloned, sequenced, assigned the spectra, got all that information, and eventually proved. And before that, we made a hypothesis that what are the detailed structural and functional changes that induce upon this mutation, and if any, and that causes such drastic phenotype in a lifetime. The hypothesis we proposed was that remarkable structural instability due to this point mutation, G to W, the glycine to tryptophan, may propagate cooperatively and sequentially through the whole structure, providing insights into the mechanism of cataract formation. And it indeed, biophysical studies already gave some information about how the stability of the protein decreases by a simple single point mutation. And cutting short the story, we did all the assignments and we looked at the beta sheet rich formation of this structure and studied the structure. And you see here three-dimensional structure of this gamma S protein, crystalline protein, where you see this tryptophan is now exposed. All those in the audience who have little information about these various amino acid residues, tryptophan is supposed to be hydrophobic because of its indole ring. This is called indole ring of the, okay? This hydrophobicity, in principle, should be buried. It should hate water. So when you dissolve it in water, it should have gone inside, deep inside the protein pocket. But unlike all the other tryptophans that are present here, this mutated amino acid residues, this tryptophan is exposed to the solvent, and that a hydrophobic residue exposing itself is the cause of the disease. And that is what we have shown. And usually when you study the structure of a protein, you come up with various statistics to show that what you have done is very, very highly resolved, high resolution structure. And we concluded that the unusual exposure of the tryptophan results in readjustment of the N-terminal domain in the human gamma S protein. And as you have seen, the polypeptide chain around this 57 is completely disturbed now. Okay? And this is exemplified even in the NMR spectrum, where you see that the signal domain is unaltered in going from wild type protein, whereas because of this single point mutation, is a lot of disturbance in the chemical shifts itself. So that itself is a signature how this is resulting in a instability in this protein in the N-terminal domain, okay? And that is eventually causing the disease. So biophysical characterization revealed structural instability. Complete sequence specific resonance instrument was done. Three-dimensional structure was determined with an RMSD of 0.5 angstroms, and tryptophan is shown unusually exposed, and this loop is affected, and conformational dynamics reveals higher flexibility of the N-terminal domain as compared to the C-terminal domain. And we have done several other studies further to highlight about all these observations, which I would skip now because of the time. And we have reframed our conclusions by saying that remarkable structural instability due to this point mutation propagates sequentially through the entire N-terminal domain, leaving the C-terminal domain unperturbed. And the mechanism that we proposed is that evidence of partially open conformations may act as nucleation centers for their aggregation. The resulting instabilization attenuates intercrystalline interactions with striking therapeutic consequences. So this is what the story of the third protein which I wanted to highlight. In the given time, I'm sure I have tried to see how you should address your questions and how you can get answers from this esoteric NMR spectroscopic technique, and which has become very powerful, not only in studying very ordered structures, but even disordered structures. Always remember that today, even disordered proteins have been shown to be much more much more important than ordered structures actually. Many a time, these disordered structures have been shown to be much more dangerous than ordered structures. So I conclude here that with a take-home message, unraveling the solution conformation of biologically and chemically important molecules, mind you, not amenable to crystallography, and studying their dynamics and interaction with other molecules to understand the structure and function relationship is a challenge. If you can crystallize, please go to crystallography lab. But if you cannot crystallize easily, always come to NMR lab. And it is there and where NMR is an indispensable tool. It is fun doing 5-day experiments or 1-day experiment or T20 experiments. It is an amalgamation of different fast techniques, and which can hand in hand go with other biophysical techniques in the process. Okay? So before I conclude, though I have talked about only a part of this esoteric technique, I want to highlight that it is right at the intersection of these beautifully growing gardens of physics and chemistry and biology and medicine. And that is what it puts all these 4 different kinds of people together. And it is proud to be an NMR spectroscopist. So all the young in the audience have some interest, show some interest, and try to experiment and see how you too can contribute to this field and take it further. And all this, what I shared with you, could not have been possible without the contributions of all my postdocs, my doctoral students and postdocs and other collaborators. Okay? And what highlighted in the red are those who have contributed to these 3 proteins which I highlighted. And without their contribution, I would not have been in a position to talk to you. And when I left GFR, and what we had was these 4 spectrometers in Bombay and another 2 spectrometers in Hyderabad that made me what I am today. And this is the book rather which I have referred to you. And I would now, after this workshop, if you can have an access to this, it has 14 chapters. It highlights even MRI, solid state NMR, metabolomics, and other things also, which I couldn't spend much time because of the paucity of time. I would strongly refer this 14 chapter, 500 odd pages book, a Springer edition, which I co-authored Professor Nobel. And thank you for your kind attention. So this is a bird, a picture taken in our new campus that's coming up. And I extend warm welcome to all of you. Okay, you can, all the young can think of coming to us and carrying out their research at our place. And we are soon going to have a large NMR facility. We have ordered a 400 megahertz NMR and 700 megahertz NMR spectrometer, and they're going to be installed very soon. 400 is already installed, 700 is going to be installed in another few months. I mean, unfortunately, this COVID is preventing its installation. But in a month or 2, I think we should have 700 also working in that. So with that note, I thank you for your kind attention, and I'm sorry if I have taken more. I was told that it is 2-hour lecture program. So that is how I put a little more extra slides, but I'm sure you all have understood. If you have any queries, please come back.
So,
Thank you, sir. Sunil, carry on.
Yeah, thank you very much for the very, very informative talk.
Thank you.
Yeah, now this presentation is open for discussion.
Yeah,
If someone
please.
wants to ask questions, please do that. You can post your questions in the chat box also.
There is one question from Mr. Avinash. What is non-selective RF pulse? Somebody wrote that.
Non-selective RF pulse is the pulse rather which excites all the spins present in the sample. Okay, a hard pulse, a 90-degree hard pulse is usually a non-selective pulse. Okay,
Right.
so it can excite all the resonances. Okay, you can also have selective pulse where, I mean, the hard pulse, which is the 10 microseconds, now has to be made to say 100 microseconds or 200 microseconds.
I see.
Yeah.
There is one question from Dr. Anidipti. What solvents are used or are 3D triple resonance? 1 minute, the question is going up. Experiments done in solid state. The second question is, what happens to the crystalline protein during cataract formation? Is the progress of cataract based on the protein mutation alone?
So what is happening, this exposed tryptophan is hydrophobic. Now, somehow it will try to hide itself. The only way it can hide is that partner with another tryptophan which is exposed and now come closer. So what it means is that 2 tryptophans of 2 adjacent molecules may come together to form an aggregation. So that nucleates the aggregation, and that aggregation process is eventually is non-crystalline, and that non-crystalline aggregation results in the cataract. And that is the story rather which we brought out of this study.
Okay, very good. I think Anidipti understood.
Yeah.
Yes, sir. Right, sir. So, sir, is that alone for that child, the infant case, or is it the normal cataract for the old people also? It's the same way.
Say again. What is the question?
Sir, is that only for the infant case, infant cataract, or is it for the normal old age cataract also? Is it the same process that is taking place?
Yeah.
Infant or old?
Yeah. As a matter of fact, the studies have shown that it is more prevalent in infants. Okay.
Okay.
By birth itself, I mean, within few months of birth itself, I mean, they develop this cataract, unfortunately. Okay.
Okay, sir.
Yeah.
Okay. Thank you, sir. Thank you.
Yeah. It is not age related.
Okay.
Of course, it is indeed, it is aging only, but aging in few months after birth itself. That's the sad part of it. Yeah.
Okay, sir.
So-
Thank you.
There is one more question from Srirashmi.
Yeah.
Role of NMR in microbiology field.
Role of NMR in
Microbiology. Biology. Microbiology.
Yeah. What exactly is your problem? Can you just explain?
So,
I guess she may be interested in how NMR can be used in microbiology research.
it's a general
question, I guess.
You see, I mean, in microbiology, for example, I mean, if you want to study, say, metabonomics, how given a metabolite, how it is getting digested, you can, in principle, study it very, very carefully. Okay? And you can see how, for example, a glucose transforms into different metabolites. So that's one thing what you can do that. Okay? And there is another very important, interesting thing that is what is coming up is what is known as in-cell Nmr. So what all we have been, what all I have shared with you is all in vitro. What you do, you biosynthetically express the protein, extract from the cells, and make a very homogeneous sample, put it in an NMR tube and study that. But there is also other way of doing it. You can see still being overexpressed in a cell and see how it within the cell rather what is the structure of it. I mean, people are putting even, I mean, there are several studies on what is known as in-cell NMR too. Okay, and that is much more right. I mean, the way to understand the significance of a protein because it is existing under natural circumstances. Okay, it has DNA, it has RNA, it has other proteins, all in its environment, and also several other cell organelles too. Okay.
But how do you subtract all other factors? When you use a live cell or something like that, because normally NMR we are using very pure compound, right?
Yeah. Usually it is oxygenated. I mean, when you keep it in NMR tube, a live cell, I mean, it has certain lifespan. So to keep it alive, I mean, you have to oxygenate and you should take care of that also. And the amount of the time that you can spend with such live sample, the in-cell samples are finite. I mean, you cannot do a 5-day experiment. So you have to do, I mean, within a span of a few hours. Maybe maximum you can extend it up to a day or so. So these are the things.
Thank you.
Did I clarify?
Yeah, I guess so. Yeah.
Is it clear?
Yeah. Yes.
Yeah.
So is there any question? Some more questions there?
Probably I can ask one general question. What about, some students may be for joining some project work. How is it possible in IISER and in your institute? Now for project
Sorry,
work, 2-3 months of project work, is it possible for the students for joining there?
It is possible, but we are open please do join. But only thing is we have to wait till this corona to disappear. Unfortunately, that is really causing havoc. And I'm sure you're all safe and healthy. And very soon we'll come out of this pandemic. And once it is done, you're most welcome to send. We are open, and we will definitely. And you can get in touch with me, and you can drop one line to us, and we facilitate local accommodation for you without any problem.
Thank you very much.
I mean, keep on your in your diary. I have said this, so please do come.
Okay, thank you.
So there are two more questions in the chat box. May I ask that?
Please, no issue. As many questions as possible.
Yeah. One, Nitin Thambi, his first question is from him. And why are deuterated solvents preferred to dissolve samples?
I'm so sorry, I didn't highlight that because of the positive time. You see, when you deuterate, the proton becomes a deuterium, okay? And because of this, the neighboring carbons or neighboring N15 has enhanced relaxation time, okay? That means the signal, the T2, is now increased, okay? So with that, the decay increases. So because the decay is increasing, naturally, the signals becomes narrower and narrower. So it is always good, particularly when you're playing with macromolecules, a very large system. It is always better to just ignore the protons, forget about them, and then completely deuterate the sample, and then you can get wonderful spectra out of that. Okay? So many a time, people do what is known as proton-less experiments. They don't consider proton at all. So they do C13, N15 correlation kind of experiments. Okay?
Yeah, thank you. So one more simple question is there. Why are deuterated solvents preferred to dissolve samples?
Sorry?
Deuterated solvents. Solvent. Why the NMR solvent is deuterated? That is another question.
Not necessarily it is deuterated, actually. To be very frank with you, in studying any protein, you do it in almost 100% water. Okay? What you add is only 5% D2, only just as to lock the signal, to lock the field. Okay? I'm sorry if I have to explain what is locking the field and other things. Inherently, any superconducting magnet has certain drift. Because of the drift, the homogeneity is not good. If the homogeneity is not good, lines becomes broad. So what you have to do, you have to make it homogeneously high field, and that is done by locking the field. And that locking the field is done by adding a small amount of deuterium. Okay? So in principle, whenever you are playing with any proteins, you take 95% water and 5% D2O. Okay? That is one question. Sometimes it may be that you don't want any external proton at all. You don't want to observe any external proton. External proton like in amide protons, okay, or OH protons, and all these things. If you are not for it, what you do, you better dissolve it in a deuterium oxide. By virtue of dissolving it in deuterium oxide, you are getting rid of all the water that is present in the sample. And because you are getting rid of the water, what happens? The way you have to do experiment becomes much easier. Because in the presence of water, you have to kill the huge amount of water, which is coming from 55 millimolar sample of water, okay? As compared to your 1 millimolar protein sample, okay? So all that water management is gone now. You don't have to worry, and the pulse sequence becomes much more simpler, much more easier. Okay. On the other hand, there are certain issues when you cannot dissolve in water a given protein or in an organic molecule. What you do? You cannot do the NMR experiment. So then, at times, in those times, people opt for some other organic solvent in which the sample, the compound or the protein or the chemical is. So it could be chloroform, it could be dimethyl sulfoxide, or sometimes it would be deuterated acetone. So there are several different kinds of organic solvents also, okay, that they are used only when your sample is not soluble in water. Okay?
Thank you very much. So, one more simple question. Can artificial intelligence be employed to solve the complexities that is present now in NMR? Artificial intelligence. Yeah.
Artificial intelligence. Yeah, it's a very interesting question actually.
Yeah. It's a question from Vincent Joseph. Yeah.
Yeah. As a matter of fact, we are going to be driven into the direction. As I've highlighted, in analyzing complex triple resonance spectra, it is time consuming and manually doing is laborious. So that is the time when your artificial intelligence, basically automating an assignment of a spectrum, is nothing but using your artificial intelligence. What exactly you're doing? You know that alanine CH3 proton resonates between 14 to 24 ppm. So that is the artificial intelligence, am I right?
Yeah.
So using these signatures, possible signatures of various amino acid residues, in principle, you can use. So when I said that software development or hardware development, I mean, as a matter of fact, I could have written artificial intelligence there. It is artificial intelligence rather which enables you to automate something. Okay?
Yeah.
So it is possible. There will be, I mean, artificial intelligence is going to enter each and every field of you. Okay, you have to live with it.
Thank you.
Yeah. Yeah.
So, more questions there?
No, I think no. So probably we can conclude right now.
I just wanted to ask my own question actually, whether I was audible to every one of you or not.
It was perfect. You know, sometimes there was some glitches, but it was perfect. It was clear. Yeah.
Okay.
Yeah.
Wonderful. Thank you so much.
Yeah. The presentation was very, very good. Yeah.
Thank you very much, Professor. It was very nice. Yeah.
It's my pleasure, and I wish all the very best in the rest of the lectures too. And I have seen you have really put very nice people on board, and I'm sure they will take you much more deeper too.
Yeah,
okay.
Yeah, sure. Yeah, we hope so. Yeah.
And the complexity too.
Thank you very much, Professor.
Thank you.
Okay,
Thank you.
all the very best to you.
Yeah, thank you. Thank you.
Yeah. Bye.
Thank you, sir.
Bye.